translation, process of formation of protein from mRNA. Now remember, we have already learned about the transcription, which was formation of mRNA from DNA. So translation is a combined process, a complex process which takes place in cytoplasm. The first step is loading of tRNA. See, this is a tRNA, which we also call it as transfer RNA. It is getting added with the amino acids. Specific tRNA binds to specific amino acid at its 3' end. With the help of enzyme amino acyl synthetase. Now you will find that tRNA with amino acid, we call it as loading. This process requires energy, which is provided by ATP. Once that is done, now next structure what we require for translation is mRNA. You know the direction of mRNA, we go for 5' to 3'. The segment of mRNA which will not produce protein is called as UTR, untranslation region. This is a translation segment. It has a start codon. You will find it also has a stop codon. Now what happens? Smaller unit of ribosome. It comes and binds to the mRNA. Smaller unit of ribosome binds to mRNA. mRNA has a site for binding of smaller unit of ribosome. The larger unit of ribosome binds to the smaller unit of ribosome. Now, because of this binding, what happens here? You will find that two set of codon are within the ribosome. Yes, these are referred as A site, P site, and E site, as shown in the diagram. A, amino acid site, P, peptide site, and E is the exit site. Clear? This process of mRNA getting arranged in this fashion, we call it as initiation process. Once the initiation is completed, next step is elongation. See here, when the initiation takes place, at the start codon, you will find a tRNA which will carry an amino acid called methionine, because for AUG codon, there is methionine. If it is a prokaryotic, you can find N-formyl methionine, and if it is eukaryotic, you will find methionine. So remember, first tRNA always enters P site. Now, you will find that there is another tRNA which had entered A site now. So now A and P both have a one tRNA with one amino acid each. This amino acid is dependent on codon which is present on DNA. Now, if both are formed, what happens? The tRNA which was present at A site transfer its amino acid to tRNA present on A site. So see here, this tRNA now has two amino acids. Once it is done, ribosome will slide over. See here, it's sliding. So when it slides, the process is called as translocation. Now, because of translocation, you will find that start codon had come to E site. This was the A site, this was the P site, and now it had become E site. Initially, start codon was at P site. Now, because of translocation, it had moved to E site. What happens here? The tRNA which was there, one new tRNA enters at the A site with one amino acid. Now, the start codon which tRNA was there, it moves out. Now, next is the tRNA present at P site transfer its amino acid to the tRNA present at A site. Again, translocation take place. You will find that now the tRNA at P site has 3 amino acid. A new tRNA will enter. The moment it happens, it passes the amino acid chain to the A. Again, translocation will take place and this process continues. This process continues till the time you will find that ribosome had moved to a stop codon. UAG, UGA, UAA. These are stop codon. The moment stop codon enters the A site, what happens? It triggers a dissociation, leaving behind the chain of amino acid. This chain of amino acid is polypeptide chain, which is called as protein.